Sorry about that. So, Mr. Chairman, ladies and gentlemen, it is a pleasure to talk about cardiac contractility. I have to start that why to study cardiovascular science or cardiovascular physiology in general. Well, it is really easy because we are human living beings, and therefore we are expected to die. And the question is that which profession is the worst? Because the worst profession will have the highest rate of death at the end of the day. And nowadays it appears that it is the cardiovascular researchers who are doing not a good job, because most of the individuals are dying because of cardiovascular diseases. So let me talk about the circulation in general. Well, the circulation consists of the heart. The heart generates of the pressure in the circulation. And after that, this heart pumps the blood into the arteries. And the arteries are supplying of the blood and nutrients to the tissues. Well, there is an important aspect here, that these arteries are not just some kind of tubes. These arteries has a regulated diameter. So if one of the tissue requires a bit more of the oxygen or nutrients, then these arteries are able to dilate and provide more blood to this tissue. So blood distribution is regulated by the arteries. And there is another phase of the arteries, which is the heart and the arteries can be taken as some kind of pumping system, like it which works in the garden. It is important that this circulatory system has a certain volume. When the volume increases, if the arteries are, stiff a bit, then the full system will respond by elevated blood pressure levels. And in a way, arteries are also regulating of the blood pressure, because if arteries are dilated, then there is more room for the blood to fill, and therefore pressure declines. Message is, arteries are important either in providing blood to the tissues and also regulating of the blood pressure. Now, when the arteries are transported the blood to the tissues, then of course blood comes back to the heart to recirculate. And this is that which makes, of course, 4 chambers of the heart. One of them of the left side is pumping of the blood to the arteries, and this blood is being collected in the right side of the heart, where it is being pumped to the lungs to reoxygenize. So basically, this is circulation. I won't speak too much about the arteries, but I will speak a bit about the heart. As I mentioned, cardiovascular disease is one of the leading causes of death in the modern societies. In some Eastern countries, stroke is a bit more frequent, and during the COVID situation, well, even in Hungary, the COVID was a major player in terms of number of deaths. So there is the Murphy's Rule. Probably many of you already know that, which says that anything that can go wrong will go wrong. I was looking back, and it turned out that it is not really Murphy who said it first, and I really prefer that way, as it is phrased here by Augustus de Morgan, in the middle of the 19th century, and it says that the first experiment already illustrates the truth of the theory that, confirmed by practice, whatever can happen will happen if we make trials enough. Why do I like that? Because I'm a researcher. So what I am doing is that I am designing some questions. I have an idea. I try to design an experiment to prove or disprove of my idea. And this is what we call as experiment, and this is what I may call as trial. Now, the question is that, first, how many times the heart tries to do the beating, in which time something can go wrong. To put it in context, then my talk here will probably take about 1,500 heartbeats of yours. Thank you very much, devoting it to me. Now, when we calculate that, okay, that's 20 minutes, but if we calculate that how many times our heart is expected to beat, well, the first 70 years of our life, we are expecting our heart to beat about 2.5 billion times. 2.5 billion times. Well, there is much there is a high number of trials, so it may go wrong, as a matter of fact. And it is not all, because there are also risk factors which make the heart susceptible for various diseases. And these risk factors are, as a matter of fact, very often, very frequent in the population, and especially frequent in Hungary. There are some risk factors. One of them is the high cholesterol level. That's one of the major risk factors in terms of cardiological outcomes. Because cholesterol, when it is increased, tends to accumulate in the vessels, narrowing down of the vessel, therefore restricting of the circulation. And it is also giving rise to thrombotic events when a blood clot is formed in the vessel, and when it occurs in the heart, we call it as myocardial infarction. When it occurs in the lungs, we call it, in the brain, we call it as a stroke. And there are many other risk factors: high blood pressure, diabetes, smoking, obesity, physical inactivity, and also aging. Now, the combination of these 2.5 billion beats and the risk factors will result in a so-called cardiovascular continuum, which starts with risk factors, normal working heart. This is the normal heart which is contracting, but there may occur atherosclerosis, left ventricular hypertrophy, which may lead to myocardial infarction or simply of the disfigurement of the heart. Then during myocardial infarction, some of the individual will die because of electrical events, and who survive most probably will tend towards to the congestive heart failure. Talking about heart failure, there are two kinds of heart failure in this process. One of them is called as diastolic heart failure, which is the problem in diastolic heart failure is insufficient filling, because there are thick ventricular walls, and therefore the lumen of the ventricles are a bit smaller. It is harder for the heart to get filled by blood, therefore it can just pump out a limited volume of the blood. The other form is the systolic heart failure. The problem here is there are large lumens because there is an enlargement of the chambers of the heart. Nonetheless, there is less contraction, and altogether we do have again less blood pumped out from the heart. So when we have a look at on the normal heart, the volume when the heart is diastole means that its largest volume is about 140 milliliters. In systole, when it contracts, it pumps out the blood to reduce the volume to about 50 milliliters. If we take, and that's a very important parameter here, which is the ejection fraction, if we divide this systolic volume by the diastolic volume, we can tell that how many percentage of the diastolic volume was pumped out. That's more than 60%, and it means about 100 milliliters. Now, in systolic heart failure, what we have enlargement of the chambers, decline in the percentage of pumped blood, and altogether we have a lower volume of stroke volume, which is the single beat, how much blood is being pumped out. In diastolic heart failure, we have a different situation. We have somewhat less, smaller chambers, similar ejection fraction, but because of the smaller chambers, still we have a lack of blood in the circulation. What is heart failure? Heart failure is a situation when the heart cannot provide sufficient circulation, sufficient perfusion to the tissues. Well, of course, it would be nice to treat that condition. There are many drugs treating that, and I just would like to introduce them very briefly. Some drugs are acting on the cell surface. They are modulating of the level of calcium, intracellular calcium, which is important for the active myosin system to generate force. And there are drugs which can be added to the cell surface receptors, which are modulating calcium. There are drugs which are modulating of the calcium interaction with the octomyosin system. And there are drugs which are acting directly on the myosin. These are the old drugs. I will talk about them, and I will concentrate on my talk about the novel drug acting on the myosin to improve myosin ATPase activity, their force production. So what these drugs are doing? When we use the cell surface drugs, one of them or a set of them will limit of the heart rates in a minute. So the heart will slow down, which will help the heart. Other drugs are reducing of the volume of the blood. And that's good because the heart does not need to move that much blood. And there are other drugs, and my talk will be about them, when we are just trying to use this carrot and try to motivate this animal to move. And these are the positive inotropic agents, which would presumably help the heart to contract. Okay, now the first experiment what we did is that we glued a single cardiomyocyte to two needles and measured each force generation in different calcium-containing solutions. So when calcium is added, this single size preparation contracted. And when the calcium was removed, it is just relaxed back. What we see, what we tested is the omecamtiv mecarbil, a novel myosin activator, and these are the results of ours. As you see, this is the calcium concentration, tricky range, because this is the highest, this is the lowest. And what we can see is that when we added a lot of drug, we can see a reduction in contractile force. Not what we expected. We wanted to provide increased contraction to these cells. Nonetheless, the problem is that we don't really know in what range physiologically of the calcium is changing here. I will show you that we try to solve that problem, because when the calcium concentration is relatively slow, we can see an increased contractility in the presence of the drug. When the drug is present, that contractility in the control increases here or here. So that's five-fold increase in this range. Problem is that we actually know calcium concentrations. This drug also increased the contractility at that time, and then even today we don't really understand what happens. So what we did is that we designed a new experimental condition when we measured the intracellular calcium concentration and the contraction of the cells simultaneously. This is the experiment, the screen of the instrument. You can see that this is the cardiac myocyte. It is contracting. This is an isolated left ventricular cardiac myocyte. This is what it does in our body. And we measured parallel of the intracellular calcium concentration here and the sarcomer length, which is proportional by the shortening. So how the result look like? When we stimulated the cell, it shortened, that's the contraction here, then it relaxed back spontaneously. In parallel, we recorded of the calcium concentration, which just raised, then get relaxed. What can we do with this figure? This is what I like the most, that since the contraction, as I mentioned, is a function of the intracellular calcium concentration, we can plot the contraction as a function of the intracellular calcium concentration. How did it start? Cell is here, then intracellular calcium increases, then the cell contracts, then the intracellular calcium decreases, then the cell relaxes. This is how it is doing that. Now, let's see one of the examples, isoprotenerol, which activates the upstream pathway. What happens when we add isoprotenerol to the cell? What we can see is that there is an increase in calcium concentration, and there is some increase in the contractility. So it contracts a bit more, much higher calcium. It is not optimal for the heart. We don't really like when calcium concentration increases because it causes arrhythmia, and energetically it is not as good. There is another drug acting on facilitating of the interaction between the actimyosin system and calcium. What can we see? No change in calcium concentration, but increased contractility. Bingo, this is what we want. Another drug, which acts on the myosin, does a bit different. There is no change in calcium. This is what we like. There is much contraction. This is also what we want or like. But I would like to pinpoint here that there is a decline in the resting sarcomer length. So it shrinks the cell. We don't really know what its effect is, but it is more prominent using another drug of the same group, omecamtiv mecarbil. You can see no change in calcium, basically. There is a much more shrinked, much shorter wavelengths at in the presence of the drug. And we can see similar features in presence of dunicumab and other drug acting on the myosin. Now what happens? This is the resting state of the cell. We are adding increasing concentrations of the omecamtiv mecarbil. This is the calcium concentration. Resting cell, it is not stimulated. What can we see? No change whatsoever. Nonetheless, the cell is contracting without calcium. Well, that was some novelty. That's why this paper has got accepted, because nobody expected that. We expected increased calcium, then contractility. Then what happens here? We don't really know. Nonetheless, we wanted to see that what is physiological effect. Here is, you can see an echocardiographic recording, as a matter of fact, my own heart. And you can observe that this is the left ventricle of my heart. And please just consider the left ventricle of the heart, because we can measure all of the characteristics in systole when it contracts and in diastole when it relaxes. When we do that, I have already talked about the ejection fraction. We saw that omecamtiv mecarbil increased the ejection fraction. Very good. This is a target for positive inotropic agents to increase cardiac contractility. How did it do? Systolic, when the heart contracted, there is less volume remaining during contraction in the heart, which means that it pumps out more volume from the heart. Good. Problem was that in diastolic stage, when we want to fill up the heart with blood as much as possible, we were also being to see a decline. This is what we didn't like, because we want to have as big heart as possible and as much contraction as possible. Now what we are seeing is that the heart is getting smaller. Yes, it is contracting more, but it starts from a smaller state. The next thing is that we applied intra-cardiac pressure and volume recording. So where does the full cycle start? Heart start to contract. It is contracting, the pressure is increasing for a while. After that, valve is being open, and this is the ejection when the blood is being pumped in the aorta. Then the pressure reaches the same level as the aorta, pump closes, heart relaxes, heart is filled up by blood. So this is basically the pressure volume curve, what we recorded. Now, same parameter, ejection fraction. We can see increase. We like it. This is positive inotropy. That was the goal. We can see that the systolic volume declined. We like it very much, because that means that in systole there is higher contraction. Problem is that in diastolic volume, the volume of the heart before contraction declines as well. And now this technique is good to calculate that how much blood is being pumped out from the heart. And it turns out that these two events, increased contractility together with decreased initial size of the heart, just knows each other. So we have a very nice parameter, ejection fraction increased. Nonetheless, we don't have more blood to be pumped out by these hearts, because they are shrinking, being smaller. And when we have a look at on another parameter, which is just correlating with the ability of the heart to be filled by blood, which is the intraventricular pressure of the blood when it is getting filling, then what we see is that there is an increased pressure here. And it is important because it appears that the heart cannot be filled properly. That's the reason why these hearts are getting smaller. There is a much stiffer heart which does not let itself to be filled by blood. Therefore, it cannot pump out as much blood as it was able before the treatment with the drug. So to summarize my speech, then I was talking about direct myosin activators as drugs to treat heart failure. We have proved that it is improving of the systolic performance of the heart. It is improving of the ejection fraction, which is one of the most important parameters for clinicians. Nonetheless, we found that these drugs are reducing of the cardiac size, leading to smaller diastolic volume. They are increasing the stiffness of the heart, which leads to reduced cardiac filling. I should acknowledge most of the work has been done by two PhD students, Philip Gábor Áron and Attila Tóth. Gábor was working with me in Debrecen. Attila was working in the Semmelweis University. There were contributions from the Department of Physiology of the University of Debrecen and also of the Pharmacy and Pharmacotherapy. Thank you very much for your attention.
Thank you for the presentation and also for keeping to time. So this leaves us some time for discussion. Anyone has questions?
That was an amazing talk. Thank you very much. So my question is, are these drugs, these new drugs, already in clinical trial or not? And what's your perspective? Based on these results, is there a good future for these drugs? Or in some cases, they may be applied, but in a lot of cases, it would be contraindicated. So how these results can determine the future of these drugs in development?
Well, most of my talk was about omecamtiv mecarbil. This is an experimental drug right now. So a couple of thousands of human beings were treated with it under clinical studies. And it was proven to improve somewhat of the cardiac contractility, but it did not improve mortality. We believe that one of the problems was that it was not targeted well, because there are two kinds of heart failure patients, as I mentioned in the introduction. One of them is the diastolic, which affects the heart ability to be filled up. And if they are not keeping these patients out of treating with omecamtiv mecarbil, then this drug will make their situation worse. And therefore, it would be good to select the right patients to be treated by these drugs. I'm not saying that it is not good enough to treat patients. What I am saying is that the patient selection is not optimal yet.
It's like, what's the concentration range? You were showing us the 340-380 ratio for calcium concentration, but what's the level inside the cell? I know you haven't calibrated this in your cells, but can you give us an idea?
Of course, I can give you an idea. I cannot give you exact numbers. That's always a problem. What I can tell is that in our isolated cell system, when we measured the shortening, the calcium concentration was in the physiological range. And we know with Zoltán that why is it important? Because these dyes, what we use to detect the changes in the calcium concentration, these are ratiometric dyes. They are working in a microscope. Nonetheless, they are not giving us exact concentration values for the calcium. So I cannot tell you that what the exact concentration is. Of course, I can tell textbook numbers, but of course, I was not able to calculate the exact calcium concentrations. What we know is that when this ratio is higher, then the calcium concentration is higher. The point what I would like to make is that that's why I like the shortening system, because I don't exactly know what the calcium concentration, but I can believe that the calcium concentrations are in the same range as they are under physiological conditions. And therefore, what we are seeing in terms of the contractility, much likely to be true for the heart.
Okay, any further questions? If not, I'll hand over the microphone to my co-chair.
Thank you.